Now, the third famous antigen-presenting cell are the B-lymphocytes. We know that the immune system is an intricate network of cells, tissues, and organs that work tirelessly to protect the body from pathogens and maintain its overall health. And among the crucial players in this complex defense mechanism are B-lymphocytes, or B cells. These specialized white blood cells play a pivotal role in adaptive immunity, along with T lymphocytes. But other than this very vital role, they are also considered as the potent antigen-presenting cells. Just as macrophages and dendritic cells. If we look at the morphology of B lymphocytes, we get to know that the morphology of B lymphocytes is distinct and reflects their functional characteristics. B lymphocytes are typically small, measuring around 10 to 15 micrometers in diameter. They have a rounded or slightly indented shape with a large, round nucleus occupying most of the cell's volume. The nucleus contains the cell's genetic material, including DNA, and is responsible for regulating cellular functions. One of the defining features of B lymphocytes is the presence of specific surface receptors known as B-cell receptors, or BCRs. BCRs are membrane-bound antibodies that allow B cells to recognize and bind to specific antigens. Each B cell expresses a unique BCR on its surface, giving it the ability to respond to a particular antigen. When B lymphocytes encounter an antigen that matches their BCR, they undergo activation and morphological changes. Activated B cells become larger and more rounded, and their nucleus becomes more prominent. These cells that are formed after B cells activation and proliferation are named as plasma cells. Plasma cells are specialized and mature B cells dedicated to producing and secreting large amounts of antibodies. Morphologically, plasma cells have an expanded endoplasmic reticulum and abundant rough endoplasmic reticulum, reflecting their high protein synthesis activity. In addition to differentiating into plasma cells, some activated B lymphocytes transform into memory B cells. Memory B cells are long-lived and remember previous encounters with antigens. Morphologically, memory B cells are similar to resting B cells but have an enhanced ability to respond rapidly and efficiently to re-exposure to the same antigen. So, we can say, the primary function of B lymphocytes is to recognize foreign invaders, such as bacteria, viruses, and other pathogens, and produce specific antibodies against them. And for the first purpose, they need to act as APCs. So, let's have a look at how these B cells play a role as professional antigen-presenting cells. We know that antigen-presenting cells are a vital component of the immune system, responsible for initiating and coordinating adaptive immune responses against specific pathogens by helping in their recognition. And dendritic cells and macrophages are well known for their role as classic APCs. But B cells also play a significant part in this process. Although the main purpose of B cells is antibody production against antigens, thus playing a role in humoral immunity. But they also possess the unique ability to act as APCs. And this capability allows B cells to present antigens to T cells, thereby activating cellular immunity as well. For the efficient working of B cells as APCs, the most important component that help them in recognition of antigen are the specialized receptors on their surface that are called B cell receptors. It is estimated that a single B cell can have around 50,000 to 100,000 BCRs. The BCR is actually a membrane-bound antibody molecule, also known as immunoglobulin or Ig, that is structurally diverse due to genetic recombination during B-cell development. That enables them to recognize a wide range of antigens. Only these receptor molecules vary a little, otherwise the process of antigen uptake is almost similar in these APCs. Like for B lymphocytes, process of antigen presentation involves these traditional steps that are antigen uptake, antigen processing, then antigen presentation to T cells, and lastly co-stimulation and T cell activation, where first step is antigen uptake. And in this antigen uptake, the variable regions of BCRs mainly play the role, which are also called FB region or fragment binding region. The amino acid residues of this FB region bind with their compatible antigenic epitope. And once they find the compatible ones, they make a bond with it. And we say it as antigen has been recognized. Now, once recognized, then they engulf the pathogen or antigen through their BCRs. Alternatively, they can internalize antigens through endocytosis or receptor-mediated uptake as well. Just the same way, as I have explained already. Then after antigen uptake, they initiate antigen processing inside the cell. Once engulfed, then, the captured antigens inside the B cell are broken down into smaller peptide fragments. As we know, the proteases and other degraded enzymes degrade the captured antigen, and through several cleavage, convert them into small peptide fragments. These fragments then associate with major histocompatibility, complex class II molecules in the endosomal compartments of the B cells. And we know that MHC2 molecules are specialized cell surface proteins responsible for presenting antigens to helper T cells. The antigen fragments then bind to MHC2 molecules in the endosomes, forming the antigen MHC2 complex. Then in the next step, that is antigen presentation to T cell, the MHC2 antigen complex is transported to the cell surface, where it is displayed to helper T cells. Helper T cells keeps on wandering in search of any danger signal, so that they can help destroy it. So, once they get a danger tag over the surface of B cells via MHC molecules, they get trigger, so they detect the antigen and start taking appropriate response against it. In this case, it will initiate both cellular and humoral immune responses, as some of B cells will convert to plasma cells and start producing antibodies against the captured antigen, and the rest will be activating the helper T cells. Then comes the next step, which is co-stimulation and T cell activation. We know that, for effective T cell activation, single primary signal generated by TCR and antigen MHC complex is not enough. So, a second signal called co-stimulation is required. And in this co-stimulation, surface receptors of B cells other than BCRs play a vital role. These are usually CD proteins that helps in co-stimulation by interaction with their ligands on other immune cells. The activated B cell then expresses co-stimulatory molecules such as CD80 and CD86 on its surface. These co-stimulatory molecules CD80 and CD86 on the B cell bind to CD28 receptors on the helper T cell, providing the required co-stimulation. This interaction enhances T cell activation and proliferation, leading to the differentiation of helper T cells into effector T cells. The effector T cells, in turn, stimulate B cells to differentiate into antibody-secreting plasma cells, promoting the production of specific antibodies that neutralize the invading pathogen. So to summarize, B cells, who are well known for their role in antibody production, also serve as antigen-presenting cells, contributing significantly to adaptive immunity. By presenting antigens to helper T cells, B cells initiate and coordinate both humoral and cellular immune responses. Their versatile functions make them indispensable players in the immune system's defense against infections. So, this was all about B-lymphocytes in context of antigen-presenting cells. All in all, we have gained in-depth knowledge about all three major types of antigen-presenting cells in this video. For more such interesting videos, keep visiting sqadia.com.
Empower your future with sqadia.com. Your journey to medical mastery starts here. Delve into a world where learning has no boundaries. Engage with our comprehensive online courses, designed to enhance your understanding and refine your skills. You control your education. Learn anytime, anywhere, at your own pace. Transform complex medical concepts into engaging, understandable modules. Experience learning that is as effective as it is enjoyable. Realize your potential, reach new heights, and be ready to take on the challenges of the medical field. This is not just about education. It's about preparing you for a successful career in medicine. sqadia.com. Transforming medical education. Shaping your future.